So I'm really nervous that I need to introduce Dr. Sang, because when I received his CV, it's like 22 pages of all the honors and the position and etc. So how to introduce Dr. Sang in like 5 minutes is really challenging. So Dr. Sang graduated from Department of Botany, National Taiwan University, and nowadays very few people know about botany. But later on, Department of Botany and Department of Zoology merged into Department of Life Science. So when I talk about life science, you know which department it is. And then he moved on to Caltech and received his PhD in Caltech. And then he got his postdoc training in Yale University. Later, he came back to Taiwan and he started as an associate research fellow in the Institute of Molecular Biology, Academia Sinica. And he stayed in the same institute and promoted all the way to distinguished research fellow in Academia Sinica. And recently he moved to, he's a joint appointment and he's a distinguished research fellow and director in the Institute of Molecular and Genomic Medicine, National Health Research Institute, or NHRI. So, Dr. Sang's research, he used Drosophila as a model and studied eye development. His study has revealed like delicate molecular mechanism of several transcription factors such as homothorax, iris, eye gum, and he also revealed some interesting signaling pathway that regulates eye development, such as DPP, BMP signaling, and JAK-STAT signaling. And later on, he moved on to study glia cell. Differentiation and development of glia cell by using Drosophila as a model. Certainly, all his work are published in high-impact journal. But what I want to say is that Dr. Sang's impact is not limited in those journal papers. In fact, his impact is much more than that. I think in Chinese, he's a 开山祖师. Okay, so in English, it's just founder. I think Dr. Sang, in fact, he's the founder of the academic society in Taiwan. And he's the founder of developmental biology study in Taiwan. And he is also a founder in Drosophila research in Taiwan. And he's also a founder in developmental neurobiology or neuroscience in Taiwan. I think that's why we respect him so much. And many of the PI or professor nowadays in Taiwan, like Wang Tingfang, like Yao Jiguang, like Zhang Chunchun in Chengkong University, many professors are in fact graduates or has been studied in his lab. So his impact is not limited to himself or his own lab. He has like spread out his impact to the whole society, not only in Taiwan in the academic society, but also I think in some of the biotechnology company, he also has his impact. So without further ado, let's welcome Dr. Sang.
Can you hear me? Okay. Thank you for the invitation and very kind introduction. I'm so happy to come back here to see some old friends and so many young faces. I was an adjunct faculty here for about 30 years. Okay. So today I'll tell you a very interesting and special story. Okay. This is special because I was known for Drosophila research, but this is not about my Drosophila research. This is research without doing experiment, okay? Because I'm about to retire, I don't have any students or postdocs to do any experiment, and I don't have publication pressure. So I just take some long-standing unsolved mystery and try to read papers and figure out how to explain it. Okay? So this is very nice because I'm not constrained by money, by personnel, or by what I was trained for. Okay? I just take an interesting problem. Okay? And I found it to be very intellectually challenging and happy. So I think this is something I can do even after I retire, which will be very soon. Okay. So the story I tell you is published this year after 10 rejections from 10 journals. Okay, finally it got published. And I have two collaborators, Wu Yeling and Liao Benyang from NHRI. Okay. So I'll first tell you about this phenomenon of heterogeneity, phenotypic heterogeneity. And it's being explained by this concept of threshold effect. And I'll tell you a molecular mechanism for causing this threshold effect, that is this ultrasensitivity. And hopefully we can explain this phenomenon, and then I'll tell you what does this have to do with our own research? How can this idea affect us? So one simple example, COVID-19. Okay? The virus is everywhere. Some people get exposed and get infected. Some people get exposed but not infected. Okay? And the severity of the symptom varies from people to people. You get vaccination and its effectiveness is variable from people to people. Okay? So the same cost, but you have very different outcome in people. So this is a phenomenon in genetics called by different names. Okay? So wild type, you have a population with no phenotype, and you have a mutant that gives you complete penetrance, meaning 100% of the individual carrying this mutation all give you a phenotype. And you have incomplete penetrance that not everybody has this phenotype. So in fly genetics, we see a lethality that's 70%. Okay? So it's incomplete. Then there's this variable expressivity. Expressivity is that you have a phenotype, but the severity of the phenotype varies. It can be very mild or can be very strong. Okay? So variable expressivity here demonstrated by the different color shades. And you can have a combination of incomplete penetrance and variable expressivity. Okay. There's another phenomenon called pleiotropy. That is a mutation will affect multiple phenotypes, multiple tissues, multiple organs. Okay. And in different individuals, maybe one person have the mutation, have the liver defect, another will have a heart defect. So this is called pleiotropy. Okay? So here the color is only partial. So in general, when we encounter these, when we do experiment with a model organism, we think this is a noise. Okay? It's a troublesome. We try to ignore. Okay? I used to say, I will work on a mutation if the penetrance is very high. If it's 20%, I don't want to work on it. Okay? It's very difficult to work on. Okay? And human genetics, human disease, there are many of these phenotypes. Okay? So I will want to stress that these noise actually carry some value. Okay? They could be very useful. Okay? So I'll give you some example. BRCA1 and 2 gives you breast cancer or ovarian cancer. Okay? But people carrying the BRCA1 or 2 mutation only have certain percentage of getting this breast tumor. Okay. And the percentage is different for the same mutation, but different for breast and ovarian cancer. Okay. On the same person, if you have breast cancer on one side, then the chance of getting a contralateral cancer on the other side is also low, okay? Not 100%. Okay? So in carrier of this mutation, every cell in your body have the mutation, but only some cell, very small number of cells, will develop into a tumor. Another example, this is neurofibromatosis NF1. For the same mutation, okay, this person, every cell has a mutation, but some cells would develop into these nodules. And you can see that some nodules are bigger, some are smaller. Perhaps this indicates a difference in onset time. Okay, so lots of heterogeneity. Another example is this famous winless mutant. Okay. It's called winless because the mutation gives you no wing. Okay. But the original phenotype affect not 100%. Okay. This is one example. Only one wing is missing on the same individual. Okay. And this is for the population that the penetrance is not 100%. Another example, this is a polymerase gamma mutation, the same mutation in different family, different individual. You can see that the clinical presentation are different. Okay? I won't go into the detail, but these are different. The age of onset is different. The symptom affects multiple tissues, and they are different. Another example, this is Bart-Pumphrey syndrome. And these are all carrying mutation, and they are different phenotypes. You can see different individuals have different combinations of the phenotype. Okay. So this raises many interesting questions. Why some carriers do not develop the disease phenotype? Okay? Why some carriers have a weaker or stronger phenotype? Why they have different onset time? Why only some tissue are affected and only some cells are affected? So why should we care? Because if we can understand, okay, we may be able to manipulate and give you a better prediction or treatment. Another example is in neurobiology. Okay, this is a famous experiment from Seymour Benzer. He puts the fly, Drosophila, into a tube. Okay. You bang the tube, all the flies drop to the end, to the bottom, and you let them climb up. Okay. So the flies will come up for maybe 30 seconds or 1 minute. Then you separate the tube. Okay. So this part are the flies that responded to the light or negative geotaxis. Okay. So you have two populations. One go up, one stay down. Okay. You tap both populations to the bottom and retest them again. Now you see that the same population that was up, again distributed in a different proportion. There's still some go up, but some at the bottom. Okay. So this suggests that in multiple testing, in every test, every individual has a probability of responding. Okay. It's not 100%. There's a probability. So many of our own behavior is something like this. So ideally, we think if you have a gene, in wild type you have 100% dosage, and gradually in mutant you decrease, and all individuals in the population will have a progressively severe phenotype. Okay? This is linear response to the dosage of the gene. Uniform, linear, deterministic. In fact, what we see is something like this. So it's heterogeneous, nonlinear, and probabilistic. So how do we explain this? Okay, many explanations have been proposed and demonstrated. So you can have differences between individuals. We have different genetic background. We have different environmental exposure, different immune history. Many of these things, okay, they all contribute. But as I said, even in the same individual, we have differences. Okay? One wing will be affected while the other is not. Okay? So this will exclude all the environmental and genetic factors. Okay? There's still these differences within a single individual. So stochastic variation, okay, noise, may be important. And there should be a threshold effect for things to happen. Okay. So you can have many different reasons giving you quantitative variation of critical factors, and this plus noise and threshold effect would explain the heterogeneous, nonlinear, probabilistic characteristic of genotype-phenotype relationship. So if we imagine a population, okay, this is wild type, and this is x-axis is concentration or activity of a certain factor. Okay. So wild type, you have a distribution like this. Mutant, you have a distribution like this. And heterozygous, a distribution like this. Imagine a threshold here. These individuals would exhibit a phenotype, detectable phenotype. These individuals will be phenotypically normal. Okay? So this determines the penetrance. We can shift the penetrance. We can shift the threshold, and this will change the penetrance. So threshold idea has been proposed many times. Okay. But what's the molecular mechanism? Okay. So people have also shown this as a sigmoidal curve as a molecular mechanism. So this is a sigmoidal curve. You see, at this region, the change in the x doesn't give you any change in the output. Okay. So at this region, it's like a wild type. There's no phenotype, and it's insensitive to any perturbation. And when you have a mutant, a null mutation, no product, no gene, okay, this gives you complete penetrance. And at this range, changes in the input gives you no change in the output. But within this region, very small change in the input will give you very large change in the output. So in this region, it's very sensitive to the change, hypersensitivity or ultrasensitivity. So you have a sharp transition once a threshold value is passed. You go from here to here. And near this inflection point, a small input gives you a big change in the output. So this system is sensitive to stochastic variation, and because this is noise, it's probabilistic. So in a deterministic situation, a fixed concentration or activity of X will give you a fixed output. Okay. But within stochastic variation, you get a range of the variation. So molecularly, we have examples that gives you this sigmoidal curve, S curve. The most famous example is allosteric positive cooperativity. Okay. So you have multiple binding. Okay. The first binding is difficult, but once the first is bound, the second, the third will be progressively more easy. Okay. So this is very common in biochemistry. So basically, you need two things. You have to propagate the effect from one site to another site, from one molecule to another molecule, or from one phosphorylation site to another phosphorylation site on the same protein. And this propagation is usually enhancing. Okay? One site will affect the other site by enhancing the activity or affinity. There are actually many more of these molecular mechanisms that gives you this S curve. These are called ultra-sensitive response motif. For example, positive cooperative binding, even negative cooperative binding, homo-multimerization, multi-step signaling, molecular titration by inhibitor, or covalent modification, for example, phosphorylation and dephosphorylation, positive feedback, reciprocal regulation. So actually we see these kind of motifs very often. Okay. And these, under certain parameters, will give you ultra-sensitive response. And this S-curve can be manipulated. The famous example is the hemoglobin binding to oxygen and CO2 and CO. So see, this is the normal curve. You can change the pH or temperature or other concentration and shift the curve to the left or to the right. And by shifting, we can change the threshold and change the penetrance. And the threshold can also change in different time. So you can have a temporal window where the system has a threshold. Okay? So you can easily imagine within this window, temporal window, certain factor is present. Okay? Other than this window, the factor is not expressed. And this factor will create this property. So the threshold can be context dependent, tissue specific, or time dependent. And this context is very important. Different contexts may mean different interacting partners. Okay? Proteins have interacting proteins. Okay? So different contexts or tissues have different interacting partners. One gene can have multiple interacting partners, give you multiple functions. So here is one example. This is me at home. I have father and mother. As time goes by, only my mother retains. So the interaction partner changes. This is my original home. Then I get married, I have a spouse, and as time changes, I have son and daughter. Okay. Your interacting partners change. Okay. Your functions change. In school, I used to have teachers and classmates, and later on, the teachers change, the classmates change. In office, I have differences. Okay. So interacting partners can change over time. Relationships, meaning network topology, among the partners can change over time. Okay. So every protein, every gene must function within different contexts. Okay. We always have to remember many things are context dependent. So basically, to get a threshold effect, I think there's 3 conditions. One is that embedded in your gene, in your genetic structure, you need to have this ultra-sensitivity module. A regulate B, you have a positive feedback. This is normally very stable homeostasis, because normally we're fine. We're insensitive to small perturbations. And these modules are tissue specific, and this is same for everyone because they are in our genes, in our gene structures. Okay. Then you may have a mutation, a driver mutation, a primary mutation in a gene, and this mutation will be specific for different people. And this mutation will shift the stable state into an ultra-sensitive window. Now you're in this part of the curve. Then the third condition is you get stochastic variation. So the X is not so stable, it changes a little bit. Stochastic variation in X or in another gene that affects X. So combining all these 3 will give you a heterogeneous phenotypic output. So one example I found in the literature. So this is in C. elegans, intestinal development. You have this kind of network, and this L2 is positive feedback. So in a SCIN1 mutant, you don't have this gene, and this gene regulates these other genes, so these are all gone. And this N1 is regulated by SCIN1, but also by another gene. So N1 is still expressed. But now the upstream regulator for L2 expression is reduced. It has only 1. So L2 is lower, and the positive feedback may not be kicked in. So now you have an ultra-sensitive situation. And sometimes the level is high enough to give you a proper development. Sometimes it's too low, and you don't have the intestinal development. So this is ultra-sensitive response motif, this positive feedback. And the driver mutation, the SCIN1, and stochastic variation in this or in this. So combining all these, you have an incomplete penetrance for this intestinal phenotype. So how does this differ from what we normally think? So let's take this example. You have a tumor suppressor germline mutation, for example, BRCA1 or P53. Every cell has this mutation. But only certain cell gives you a tumor. Traditionally, the idea is that you need to have a second site mutation. And this happens stochastically, random, at a low frequency. So some cell will develop a second site mutation, and that will cause the phenotype. But the threshold model will say that you don't need to have a mutation. It can just be a stochastic variation of the gene level. And that will, below a critical threshold, will give you the phenotype. So what this suggests is that when you have a tumor, you look for, by sequencing, to look for a mutation. You may not find it, because this other thing is not a mutation, but a stochastic changes. Not all tissues are affected. Okay? The traditional way is that certain limiting component is tissue specific in their expression level. Okay? And I always argue that the certain limiting component has tissue specific threshold rather than the level. The threshold can change in different tissues. Okay? the level may be the same. And also the onset time, okay, again, it can be the time you take to get a second site mutation. So sometimes it's very long, sometimes it's very quick. Or even the mutation rate. And the threshold model will say that it may be the level of noise or the level, the threshold level can change at different age. So these will give you different interpretation and should affect how you look for the factors. Another important thing is this pleiotropy. Okay? A single gene affecting multiple tissue have multiple functions. We can imagine this kind of network. These may be protein interactions or gene interactions. Okay? So one gene will connect to other things. Okay? This is called a node, and this interaction is called an edge. Okay? If you remove a gene or protein, okay, you lose all these connections. Okay? So none of these will be functional, and this is total loss of function. Or you can just reduce the level of activity, and this is a partial loss of function. Okay. But you can also affect only one edge. So this protein is normal, but it does not link to this part. So this edge. And we can imagine pleiotropism is something like this. This gene mutation affects only a certain function, certain interactions. Of course, you can also have a gain of function that interacts with something new. Okay? This is much more rare. This is very common. There are many estimations that a huge proportion of mutations are genetic mutations. So everything I talked about is threshold effect at the cell level. We can easily imagine the network of regulation within a cell. Okay. Does the same thing happen at the organ level or the organismic level? Does the same regulatory network apply for interactions among cells within an organ? Do similar threshold or ultrasensory response also operate at the organ level? For example, organ failure, like kidney failure or heart failure. And does a function of organ require cell-cell interactions? For example, positive feedback is an ultrasensory module. And we see a lot of positive feedback among cells. Okay? So maybe this is a mechanism. So try to find certain potential examples. One is this Drosophila mutant called drop dead. This is the wild type brain section, and this is the mutant brain section. It's called drop dead because it functions normally. It walks, everything seems normal. Then suddenly it will drop and die. So if you take this fly and dissect the brain, you see a lot of empty holes, vacuoles. Okay. So before it dropped dead, the fly actually has a lot of neurodegeneration, but it can maintain its normal level of function until a snap, a threshold is reached, and it dies. So maybe this is one example. Another example is a behavior response. You make a choice between different odors or between light and dark. And every time you make the decision, it's a probability. Death. When we have a mutant, we very often draw a survival curve or tumor. Okay. The patients have a survival curve. Okay. It has the same condition, but at different time point, this individual will just die. Okay? So this may be another probabilistic at the individual level. Okay? Or heart failure, okay? Sudden cardiac arrest. My favorite way to die is to die very quickly, suddenly. Okay? How does this heart, the whole organ, suddenly stop beating? Okay? Another potential example is Parkinson's disease. Okay? Parkinson's disease affects the dopamine neuron. And it happens in two different systems. This is more famous, the motor symptom as in the brain. Okay. And you have a progressive loss of dopaminergic neurons, okay, until you reach 70% loss. Then you start to have this motor phenotype. But it also affects the autonomic and enteric nervous system. And 20% loss will give you a phenotype. So the different systems have different thresholds for developing the phenotype. Okay. And one explanation is that in the brain, all these neurons are highly connected, so they're more resistant to some loss. Okay. The remaining can still compensate. Okay. So the threshold is high. And in the gut, it's less connected, so more sensitive to the loss. So positive feedback happens very often. In many situations, we have this mechanism called quorum sensing. Okay. So in bacteria or in slime mode, each individual secretes some factor outside. And if everybody secretes, then the concentration extracellular will be high. And this will tell the cell that we have a high population density, and then something happens. Okay. So this is used for many single cells. But also, there's an example by 中正明院士, mouse hair follicle regeneration. Okay. If you injure the skin, if it's a very small injury, the hairs will not regenerate. But if it's a larger region, many hairs are affected, then there's a quorum sensing mechanism, and the hairs will regenerate. Okay. So maybe this kind of mechanism happen, and we just have to think in this way. Positive feedback in tumor. I looked in many tumor papers, and we can always see there's something that's positive feedback loops. Okay. But I don't know whether there's any good measurement, quantitative measurements to show there is a threshold for tumor formation. So in summary, at this part, I propose that the threshold effect is a unifying principle for phenotypic heterogeneity in human genetic diseases and in many genetic organisms. I propose that the ultra-sensitivity network module is the molecular mechanism for threshold effect. I propose 3 conditions to give you this threshold effect and emphasize on network topology and stochastic variation. So, just knowing this is nice, but does it matter? Okay. So can this really help us in our own research? So I divided this into different parts. When we study human genetic disease, we have to identify the primary mutation and then sometimes try to find the modifier factors, other genes that interact. We need to have functional validation of the gene-phenotype causal relationship. And following this model, we should identify the ultra-sensitive response motif in the regulatory network for this disease. And then we should find ways to nudge the system for hypersensitive state to a stable state. I use this word, nudge, it's gently push. Okay. So this is different from many of the therapeutic intervention that you try to block one gene completely, block one signaling completely. What I'm trying to say is that you just need to shift a little bit, and then you can have a big difference in the outcome. So, trying to identify the primary mutation, I suggest that you should pick the right target. Okay. This is a famous graph showing that the mutation allele frequency, this is very common, this is very rare, so you have rare disease. Okay. And this is the effect size, the effect of the mutation on the phenotype, and this is very high. Okay. So, very common and very strong effect. These are very easy to study. These are called low-hanging fruits and already picked, okay? Already done. Okay? There's no chance. These, very common but very small effect. Okay? Many people use GWAS to look at the population and try to find these. I would say this is not useful. It's a waste of money. Okay? If you are very rich, you can do this. Okay? For most of us, don't do this. What you want to do is look at very rare cases, but very high penetrance. Okay? And these are called rare disease. Okay? They're recognized because usually there's a family of multiple individuals carrying the same or similar phenotypes. Okay. So identification of rare diseases depend on familial cases, depend on your collaboration with clinicians who will have these patients. Okay. And they usually have very high penetrance, otherwise they could not be recognized as a familial case. Okay. So these are easier to do. And phenotypic heterogeneity provide actually important information, not just noises. Okay. Focus on familiar case, especially the extreme case, okay, because these will represent enhancer mutation or suppressor mutation. Okay. So within the family, you look at these carriers. Some carry the mutation but have no phenotype, or some have very strong phenotype, or very early onset, very late onset. These extreme cases will give you strong modifiers that can be worked on. Okay. And heterogeneity in these phenotypes suggests the system is near the threshold. So it's easier to find these modifier mutations, and you can identify the ultra-sensitive module. Okay. Pleiotropy is also useful to identify agenetic mutations. Okay. In the beginning, I showed you pleiotropy diseases. So modifier screen, okay, we usually take one example, put it in the Drosophila, and look for modifier mutations. We usually look for enhancement or suppressor of the phenotype severity. Okay, this is phenotype severity or expressivity. But I think we can also look for changes in the penetrance. You move the system from 60% penetrance to 100% penetrance. Okay. This may be a shift in the threshold. And we can look for changes in pleiotropy, okay, affecting this tissue, now affecting multiple tissues. Okay. And you have to do all of these modifier screening in a sensitized background. Okay. Sensitized background meaning you change the system to an ultra-sensitive window. So this is your window for modifier screen, sensitized background. But remember, the background is context dependent. So it's sensitized to this tissue, this condition, may not be sensitized to another condition. So ideally, we should screen or test in multiple independent genetic backgrounds. Then we usually have to do functional validation in a model system. Okay. You put the gene in the fly or mouse and try to see if you can recreate a human disease phenotype. Okay. If you can, then this demonstrates a causal relationship. But always remember that mouse or fly are different from human. So the context is different. So you put this disease gene into a fly. It may not be the same for every tissue. Okay. So if it shows there are multiple tissues, and you can only test the tissue with a similar regulatory network topology. Sometimes this tissue has no phenotype. Okay? So don't try to make a mouse model that's 100% similar to human phenotype. Okay? You may not get that. Okay? If in one tissue it can mimic the phenotype, the human phenotype, that is good. Just focus on that. So once you have these modifier mutations, you have the primary mutations, we can develop this network of regulatory interactions. Okay. And from these interactions network, there are many softwares that will help you to identify these modules, okay, with ultra-sensitive response motif. This part, I don't really know how it works, okay? But there are many people trying to do this more efficiently. Okay? And I should emphasize that just having an ultra-sensitive response motif doesn't guarantee you that will give you this kind of S-curve. Okay? It can only happen within a certain condition. For example, this phosphorylation dephosphorylation cycle. Okay. Ultra-sensitivity occurs when the catalyzing enzyme is operating near saturation. That is, the substrate concentration is high. That will give you a zero-order kinetics. And also with no product inhibition or sequestration. Okay. So these are the conditions. So for every motif, you have to check whether your condition, the concentration or everything fits, then you'll get the special character. So this requires experimental validation. And then the idea, the final goal is to change the threshold. Okay? So ideally, if you have a system, we can push the system to here, then it will be stable, okay, and insensitive to any perturbation. Okay, so how do we do that? So the idea is that the change can be very slight. Okay. If you have to use drug, the drug will only give you maybe 5% or 10% change. This way, the side effect will be low. Okay. So, for example, in this example, SKIN1 mutant, and the system is dependent on N1 or L2 level. So we can just increase the N1 level, or increase L2 level, or increase the positive feedback. This way, you will bring the system back to a stable state. Another very good potential is this protein sequestration. So very often we have an active protein binding to another protein that will inhibit its activity. Okay. Now, if you remove this inhibitor, the protein becomes active. Okay. And this is stoichiometric, one-to-one binding. So by changing the concentration of this inhibitor, we can change the behavior of this system. So a dominant negative inhibitor forming an inactive dimer, and this converts a graded transcriptional response, a linear response, into an ultra-sensitive response. So we can change the inhibitor and tune the system. So to summarize this part, I give suggestions on research strategy in identifying the primary mutations and modifier factors, and for functional validation of the causal relationship, and look for ultra-sensitive response motif, and find ways to nudge the system. Okay. So in each, I try to give some examples. Okay. So I'll just say something that noise, or the system is not very stable, it's sensitive to noise. This is actually something that may be useful for the system, for the organism. Okay. So I give you this example of cherry blossom. You have seen cherry blossom all over the mountain. Okay. Every tree blossom at the same time. Okay. The advantage of this is that if the flower requires bees to pollinate, cross-pollinate. So you open your flower, you hope other flowers also open at the same time. Okay. The bad thing is that if every flower opens at the same time, then there's a storm. Then all the flowers are gone. Okay. And this year you will have no seed. Okay. So if you observe carefully, very often not every tree will blossom at the same time. Some are late, some are early. Okay. The same variation, same 品种. Okay. Even on the same tree, not every flower is blossoming at the same time. Some are early, some are late. So this is called bed hedging. It provides diversity. So this may be inherent, built in. I want the system to be heterogeneous. To make the system very precise, it takes energy. It takes many regulatory mechanisms to keep it very precise. But there are situations where we don't want it to be precise. So now I want to expand this idea of threshold into even broader domains. Okay. So I have a discussion group in Academia Sinica. I find people in molecular biology, genetics, neurobiology, ecology, evolution, oceanography, fishery, epidemiology, social network, communication, physics, chemistry, statistics. Okay. I tell them about this idea, and everyone says that we have the same phenomenon. Okay. So the threshold phenomenon, or sometimes called a tipping point, or inflection point, or regime shift. Okay. For example, 集体合作、创新的扩散、疾病、疫情、谣言、rumor、断电、塞车、股市崩盘、social segregation、Me Too、phase transition、turbulence、avalanche、chaos. Many of these phenomena have a threshold. So this is studied in many different systems, described in different ways. Okay. So what I'm trying to see is that whether there's any generalization. Okay. Can we generalize the description or definition of the threshold? do we have the same way of presenting or representing the threshold as the S-curve? Is this a common way? Okay. And is there a general mechanism of threshold effect? Even in biology, I showed that in molecular mechanism we have multiple ways to do it. Are there any deeper similarity in these? How can we change the threshold? Is there any general mechanism? How to manipulate? Okay. And this is coming from the social science or ecology. Can we have an early warning signal for something that's close to a threshold and something is going to happen bad? So I'm hoping that combining all of these expertise, we can try to answer these questions. Okay. And one thing I learned from talking to these people or reading their paper is that there is a phenomenon called flickering. This is in the ecology. Okay. They try to predict, find an early warning. You find that when the system is close to a tipping point or close to the threshold, it can reversibly flicker. Okay. And this also happens in many diseases. Okay. People with dementia, Alzheimer's disease, sometimes they are perfectly normal. Then there are a few days that they forget everything. And then they're back to very normal. So this is reversible for a certain time and then reach an irreversible state. There are many diseases we can find this kind of situation. So maybe this is also a threshold event, and this may be a way that we can, as an early warning sign. So I think there are a lot of things that we can learn from each other in this cross-discipline discussion. Okay. So at the end, I want to tell you my experience in working on this. Okay. I think this may be useful to the teachers and the students. So I start with a question, okay, like everyone. I do reference search. So in PubMed, you can search keyword. That gives you some papers. From each paper, the PubMed will give you similar articles or cited by other articles. So from one paper, I can extend to many other papers and then extend to many other papers. So this way I can find many related papers, not necessarily have the keyword I started with. So then I downloaded for my review, I read about more than 1,000 papers. Okay. Not carefully, but at least scanned. Okay. Then you need a filing system. Okay. So I developed a filing system of so many papers. Okay. Then, I think this is important. I write something, okay, and after writing to a certain degree, I make a PowerPoint. When I use PowerPoint to present, I find ways that my text does not carry. Okay. My organization will have to change so that I can do a better presentation with individual slides, each carrying a certain message. Okay. So back and forth from the Word to PowerPoint help me to organize my thoughts. Okay. So I think this is very important. And then I talk to people. I try to present. I find my friends and I present them. And they give me suggestions, certain points they don't understand. Also, even during my presentation, I found ways that I didn't write down or didn't make clear. Okay. So this helps a lot. Then I give my mature manuscript to other friends to read and ask them to give me critical suggestions. So all of these feedbacks are very useful. So I think 2 ways to make writing or presentations better is that PowerPoint versus text, okay, back and forth, and also getting feedback from other people. And make sure you get people that can be very critical. Okay? If you get your friend says, "Oh, this is nice, nice, nice," that's not useful. Okay? You want your enemy to. Okay. So this is how I worked on this, and I hope this is useful. So I just give you one example of this way of doing research without doing experiment. And I personally found it's very interesting. I feel very good that I can finally make a paper out of it. Okay. But even if I don't publish, I still get a lot of joy, a lot of fun from this. Okay. Reading, thinking, and I think this is the essence of doing research, okay, what we originally wanted. Okay, so I'll stop here.
So now we'll take questions. So I think if you want to ask in Chinese, that's fine, or Mandarin, that's okay. If you don't, but I think it's nice that Dr. Sun always brings something new and something different. Every time I think I listen to Dr. Sun's talk, it's just refreshing. Okay, so welcome. So any question from?
Okay.
Cool. It's my second time, still a lot to digest. So I'm still trying to organize my thoughts. I'm really curious about those ultra-sensitive responsive motifs. Because for the example you gave us, usually those are the molecular interaction to underlie specificity or robustness, right? For like the collaboration between molecules or the positive feedback, for example. So it's really, I'm sure that's my soul, but would you comment on like the heterogeneity versus the robustness of the system? It sounds like the biological system use those motifs to be really strong, to be really robust, right? So almost convert the dosage into a binary system is either on or off. So it's really robust. But it's like it's the side effects, because usually they want the system to be very robust, but the side effect is now they have to deal with the heterogeneity.
Yeah, well, I think that's a very good question. I think the system is built to give you a very robust outcome, okay? And this is here. So normally this is where you are. But if you now have a mutation, this would shift the system to this state. Without the mutation, everything is very stable, very robust. With the mutation, your system may change to this ultra-sensitive region. And not every mutation will give you that, just certain things. Okay. We know some mutations give you complete penetrance, very easy to work with, but some will give you a weak phenotype.
What I try to express is, it feels like those ultra-sensitive motifs, they are not for all molecules or genes. They are for the particularly important mechanisms, because those mechanisms are really important. So that's why they have to have this feedback loop to really make sure this can go a long way, right? So, so when we see because I think for we working on genetics, the worst mutation, not just for mutations within one genes, but all the mutations for all different genes, I think the worst scenario is it doesn't cause any penetrance, right? There's just no phenotype because of redundancy or anything. So I guess what I'm trying to say is for those, it can deliver any kind of heterogeneity. Those are really important modules specifically because they are by design, they contain this feedback effect because those are important modules and they show the heterogeneity. I guess what I'm trying to say is for those, we can see the heterogeneity. Those probably like a really important pathway to begin with.
Yeah, I think many things are designed to be very stable, very robust. For example, the winless pathway, Wnt, okay? It's involved in many things, and mutations will give you many different diseases. Okay, so, but normally, it's very stable, efficiently used in multiple development process. Okay. So, for example, this winless 1 mutation I showed, it's a deletion of one small fragment containing a Wnt enhancer. So it's affecting only the Wnt expression. And it's only reducing the level to a certain amount. Okay. I think that made it to be sensitive, hypersensitivity in the Wnt, but not in any other tissue. That is why the Wnt phenotype becomes ultra sensitive and variable. Okay. But everything else is fine. 哎，同学们不要shy。
对啊，用中文也 OK 啊。当然其实我刚才听孙老师讲，我突然就想到还有某一类的 molecule， 像是 microRNA。那其实它事实上本身曾经有，其实 microRNA 很多都没有 phenotype。那可是他们事实上后来有一类的说法，说他们叫做所谓的 buffer， 就是说它事实上就是让这个系统让它避免有任何的进入那个 hypersensitive 的状态，因为它就是让所有的基因的表现都变得比较的突然增加或突然减少，都让它变得比较平缓一点，比较闹一点这样子。然后让它不会那么突然，那不会那么突然的时候，它事实上基本上它就是 guard 这个整个 system， 让它不会进入那个所谓的 hypersensitive 的 mode。对啊，只是就是想到这样子的一个。
很对，另外一个例子是 chaperone， heat shock protein， 忘了70还是90，那他们就发现这个是一个 molecular buffering。那假如你把这个拿掉，那很多隐藏的 phenotype 就会显现出来。就这个 population 其实本来可能带有很多的 mutation， 可是 phenotype 显现不出来。当这个 protein chaperone 的这个 buffering mechanism 拿掉以后，那些 phenotype 就都出来了。
I think it's a very interesting question in general in biology that Son is presenting today. So of course we have the situation that the ultra-sensitive motif, we have also the buffering motif. But if we go back to the original question that there is a phenotypic heterogeneity, is it possible that there are certain systems that were designed to be phenotypic heterogeneity, that without having any ultra-sensitive threshold, it was originally designed to be stochastic? There must be some certain example in biology that the system was designed to be totally stochastic, or somewhat stochastic, and that doesn't require ultra-sensitivity module.
Okay, so if we think the S-curve is the general representation of the threshold effect, then anything that will give you this S-curve will give you a heterogeneity in the phenotype. Okay. So maybe the system is initially just set at this window. So it's meant to give you the heterogeneity. Okay. But you're saying that is there any other things without having this kind of module, but it's already like that, then I don't know. I haven't found any examples. I think one difficult is that we have this concept, but finding real examples is difficult. Many things are not quantified very carefully. So you need to have a series of quantitation and plotted against the phenotype. I'm still trying to continuously finding these examples.
For what, I don't know whether this is a good example. For like say X chromosome inactivation, if that is completely stochastic, like which one is inactivated, that can come as some sort of like naturally built heterogeneity because every cell of this residence choice which one or the other.
Yeah. That's a good example. There, so the inactivation may start stochastically, but has to spread very quickly along the whole chromosome. So there must be some kind of spreading or positive feedback mechanism. There are many other mechanisms regulating selection between alleles in different chromosomes, something also similar to that.
Thank you, Professor. Your wonderful talk to us. Actually, this is my first lecture in this school, and also my first time to listen ultra-sensitivity threshold. My question is, when I read your latest publication, there is an example about SMA, and I work in SMA, this field. So I'm wondering how this mechanism or hypothesis can apply to the disease, and can you explain in more detail by this example?
Okay, well, I know SMA, but I don't know the details how to apply this to the disease. The disease has phenotypic heterogeneity, okay? People carrying the same level of the mutant protein, some have very severe, some have very weaker, some early onset, some late onset. So I would say that there is this threshold effect working on it, but I cannot say what is the real molecular mechanism.
I'm wondering this effect will be changed by different age in individual lifelong, or that is like a fixed mechanism?
You can imagine it changes with time. For example, if certain degradation or expression decreases or increases with age, then the whole dynamics will change. SMA is very interesting because there is multiple examples of different levels but giving you different outcome. So it mimics a threshold effect.
Just a quick comment on the stochastic probability property. So Professor Xu was saying when there is a pure stochastic process, may underlie this heterogeneity. But I think even in Dr. Song's proposed model, the noise is quite stochastic, right? It's basically the source of a stochastic. It could be from the stochastic gene expression. The level could be up and down just randomly. So that will contribute to the sigmoid model, but it's a stochastic part of the model, right? And just my thought. So the other thing is, I think the gene expression could be quite stochastic, and it will be interesting to find a way to, if we can manipulate the level of noise, it would be interesting to see how it affects the heterogeneity of the phenotypic expression.
Yeah. During development, many of the important regulators are expressed at very low molecular numbers. So there, the stochastic effect will be more pronounced. But, well, there are many mechanisms that can generate this kind of thing. And when you give a talk to advocate for this concept, I try to say that this is very important, but I want to show that this is not the only mechanism. There must be other things. This only explains the stochastic probabilistic part of that. Okay. Still, the gene will give you a certain effect that's fixed.
Any other question? So I think
孙老师，孙老师你好，我是中大所的学生。然后我有一个小小的疑问，就是老师在讲演讲的时候，主要是在探讨就是基因跟表现型、外显性的阈值的分子关系嘛。对，那我想问看的是说，有没有一种可能是我们后续可能在研究药物的开发的时候，是不是可以用老师这个模型？然后去套用在可能不只是遗传疾病上面，还是说可能用在癌症治疗啊，或是可能更多其他疾病，是不是也可以有这样子的表现型的模式？想问问看老师的。
对，这是我的希望啊。希望说，大家可以听了这个以后，很多地方就会用这样的概念去想一下。像我刚才举的例子，我自己去查，在很多的 tumor 里面，它其实是有 positive feedback loop 啊。那通常 paper 就写到有 positive feedback， 那这个也许就是一个可以去 manipulate 的点，就是你中间找到某一个点，你可以减少一点，也许就打断了它的 positive feedback， 那整个这个 tumor 就不会形成。所以也许 drug 的发展就可以去找有没有什么东西是有这样的性质。
我想都从这个问题的延伸一个问题，就是那老师这里认为说，就是这个模型会不会其实对于未来个人化医疗的建立，或是可能可以建立一个类似用这样的模型建立一个平台，做一个药物筛选的平台，有没有这样的可能？这样子。
呀，个人化医疗或者精准医疗，我这边强调的就是它是 variation， 而不一定是基因的 mutation。所以很多个人医疗是从基因的 level genomic sequence 着手，我觉得就会 miss 掉一些东西。当然，他一定会找到一些重要的，可是有些东西是那个找不到的。那然后另外一个是 context dependent， 所以你不同的 tissue、不同的这些看到的都会不一样。所以今天你假如是抽血来检验某些 metabolome 的变化的话，它可能不反映在那个 organ 那个 tissue 里面的状况，所以基本上就增加了更多的难度。
好，谢谢老师。
OK， 还有其他问题吗？那我想孙老师今天的 talk， 就是平常我们听的都是非常的 experimental biology， 就是真的是实验这样 hardcore 的那个。但孙老师今天的 talk 其实是比较在概念上一个比较 conceptually 的 talk。那我自己其实我也是第二次跟林敏杰老师一样，是第二次听这个 talk。 其实我觉得听这样的 talk 其实需要更多的时间来消化，要去事实上真的很多其实像真的需要一点经验，还有需要很多实验的经验，真的在平常实验里碰到的，然后你当然到这个时候也许就会想到。那我觉得同学就是虽然也许你们对这个东西，这是一个一个概念，那你们就是 keep in mind， 把这个概念放在心里面，就像孙老师讲的，然后把这个概念放在心里面，当你下次再看到实验结果的时候，也许有一些可以是从不同的角度、不同的来看这些实验结果。好，那我们非常谢谢孙老师今天的 talk。那我们那个院里有准备一些那个感谢状跟一些小礼物，那我就请陈君明主任来。对。